Hello everyone, my name is John Umek. I'm with the Desert Research Institute, and today I'm going to be talking about the ecological health of spring ecosystems in the western United States. So today I'm going to be following this outline. I'd like to start off by giving some general background information on springs and some of the impacts that we've seen to these springs, and then how these impacts have influenced BMI community structure in these springs, and BMI being benthic macroinvertebrates. I'm going to give a case study that's done in eastern Nevada, really close to Utah, on looking at these impacts. Then I'm also going to move into some of the work that we've done in Bryce Canyon National Park and looking at some of the same parameters as the case study that I'll be talking about, and then go into a summary and future directions for our work. So starting off with general springs background and impacts. So I'd like to start by describing some arid spring characteristics. So each spring is relatively simple, but collectively complex across landscapes. Relatively small compared to streams, rivers, or such. We can find springs individually or in large provinces, as you can see in the upper right-hand picture, and isolated from other systems. Often the only water source over the landscape, which makes these incredibly valuable ecosystems, particularly in arid landscapes. Each spring is distinctive based off of source type, whether it's a helicrene, limnocrene, rheocrene, the geology that the spring is found in, which is going to impact the geochemistry of the water. Discharge is going to play a major role, along with temperature and what we may see as far as the aquatic community. And there are a lot of other variables that are going to play into creating these distinctive springs across the landscape. So I'd like now to introduce some work by Seda and others of spring surveys conducted in the Great Basin into Utah and down all the way into the Mojave Desert, starting from the late 1980s into 2013. So the map below is showing all these different locations that have been surveyed. A quick note is that there is not a standard protocol or sampling regime that has occurred for every single location. A lot of these locations have simply been assessed either for the condition of the spring or, as some of you may know, Don Seda's focus on presence absence of Pergolopsis snails. But we wanted to take all this data, and particularly the data that did have the use or the condition of the spring, and look at it all together. So here we have a breakdown of the spring characteristics for the 2,256 springs that were surveyed. So we have a large range concerning elevation, so from minus 77 meters in Death Valley all the way up to 2,209 meters. Temperature range from 10 degrees Celsius all the way up to 39 degrees Celsius. EC from 66 to well over 60,000. Spring brook length from small springs of only 20 meters to well over 2,000 meters. And then carbon-14 residence time from 89 years to over 28,000 years. Of the 2,256 springs that were surveyed, 83% of the springs had some evidence of human use, which isn't really unexpected, but 63% had moderate or high levels of disturbance. And when we look at the BMI populations, particularly the spring snails, the Pergolopsis spring snails, we see that we have 30 extirpations from different localities and then 8 complete extinctions based off of previous surveys before the 1980s. So the effects of disturbance on these springs, when hydrology isn't a driver, when we think about environmental impacts, we see wider, shallower spring brooks. We see a decrease in energy input and current velocity. we usually see an altered thermal regime. So due to this wider, shallower channel morphology, we see an increase in temperature, and then we see a decrease in the environmental heterogeneity. Biologically, we see an alteration in the microhabitat availability. So we see changes in depth, current velocity, temperature, and then therefore an alteration in the BMI community structure in these springs. Now I'd like to move into a case study looking at the impacts to BMI community structure in springs. So we ended up collecting BMIs from 45 springs from 2 different locations in the Great Basin. When possible, we sampled at the source of the spring, 50 meters down, 50 meters below the spring source, which we called the middle, and then another 50 meters below that, which we called the lower end. Even if it extended past that 100 meters, we only collected those 3 samples at those 3 locations. We collected 5 samples at each locations and then compiled them. At those locations, we also took a lot of habitat information concerning the spring condition, the overall spring condition. Then we took velocity, wetted width, wetted depth, and then vegetation cover, substrate type, debris, and other information. SATA developed 3 categories for spring classification. So slight impacts, we have woody vegetation present and abundant. We do have ungulate hoof prints, but uncommon. But more importantly, we have vigorous riparian vegetation. Moderate, we have woody vegetation sparse or potentially even absent. We have ungulate hoof prints common. And we still have vegetation cover greater than 50% of the overall springbrook length. And then high impacts, we have woody vegetation is absent, heavy ungulate footprints, and then less than the springbrook, less than 50% of the springbrook doesn't have vegetation cover. We looked at 2 different study areas, the first one being the Black Rock Desert in the northwest of the Great Basin. So by reach, we had 15 source samples, 10 middle samples, and 12 lower samples. And then by site use, we have 3 slight, 10 moderate, and 2 high. Our other study area was the Ruby Lake National Refuge. So we had 30 source, 5 middle, and 5 lower. But here by use, we have 30 slight use, 3 moderate, and 3 high. And so we took these conditions and grouped them by reach and then by use. The first thing we looked at was spring disturbance based off of those 3 categories I just talked about versus the biological relationship. So what you're looking at here, non-metric multidimensional scaling plots, or NMDS plots, which are distant-based ordination analyses. So what we're looking for is similarities or differences within the BMI communities based off of the disturbance levels. So in red, we have slight disturbance, and in pink, we have moderate disturbance, and gray, we have high levels of disturbance. And what you can see is with just slight disturbance, we have a very compact BMI community structure. But when we start to get into moderate and high levels of disturbance, we see a lot larger range of BMI communities in those systems. And when we look at these relationships statistically, we see that there is a significant difference between slight and moderate and slight and high. However, when we look at the statistical difference between moderate and high, we don't have any significant difference, as shown with the pink and the gray areas overlapping. So our next question is, we wanted to look at these disturbance at a spring gradient scale, moving from the source to the middle to the lower section. And at moderate disturbance, we see that we have an overlap of BMI community structure, indicating that for the most part, the BMI community structure is homologous from source to lower. And there's no statistical difference based off of where you sample those BMIs concerning community structure. But when we look at slight disturbance, we do see a difference. We see at slight conditions, all of those BMI community structures are different, with a significant difference between the source and middle samples and the source and lower. And that's pretty evident looking at the figure, with each community being very separated from the other ones. However, we don't see a difference between the middle and lower source. And this could be as we start to see a mix of our tolerant and intolerant organisms found in both of those locations. Next, I'd like to talk about our work in Bryce Canyon National Park. Sampling was originally supposed to only occur in 2021. We were supposed to have 2 sampling periods. However, due to the monsoon season that year, we stopped collections after our first sampling period and then collected twice in 2022. We targeted 9 springs for each sampling period. In 2021, we collected 24 total samples from those 9 springs and identified and enumerated a little over 1,500 BMIs. In 2022, we sampled in June and August at those same 9 springs. We collected a few more samples. The 29 samples, we collected a few different springs that we didn't originally collect in June and in August in 2021, but I've actually left that data out of the analyses I'm going to show. But in 2022, we collected a little over 3,500 BMIs and identified and enumerated those. Real quick, the reason for the change in sampling periods, as you can see based off of the red marks for the figure on the left, this is the precipitation in inches for that year. And you can see, although it was really nice when we were there, we were right in the middle of the monsoon season. Whereas in 2022, we collected in June before the start, and then in August of 2022, and luckily just missed a heavy monsoon season later in September. Here is a map of our sampling locations in August 2021, June 2022, and August 2022. Our locations range from the southern end of the park all the way to the northern end of the park and included multiple elevation and gradient changes. One of the first questions we wanted to look at based off of the monsoon season that we had in 2021 and what the potential impacts were to the BMI communities is we wanted to look at the community structure by sampling period. So this is the figure A on the left, where we can see that there was no real difference between the community structure sampled in August 2021 and June 2022. However, by August 2022, we do see a statistical difference between the BMI communities, indicating that the system recovered after the monsoon season. And lastly, we looked at the community structure by gradient, source versus middle versus lower. And interestingly here, we see that there is a statistical difference between the source and lowest, but no statistical difference between the source, the middle, and then the middle and the lower sampling locations. Lastly, a quick summary and future directions. In summary, we found that it is important, when possible, to sample at multiple locations along the spring gradient to determine the overall spring community structure. At moderate and above impacts, we see a change in the BMI community structure, moving to a homologous structure. However, springs can recover to previous community structure levels after disturbance events if the intensity doesn't cross a tipping point. This leads us into future directions where we are trying to understand these tipping points for BMI communities. We are looking at indicators that may reveal impending collapses, such as hydroecological thresholds and things like that. And one of the ways we're doing that is trying to characterize historic and current food web structure using stable isotopes to understand changes in those community structures from geochemical and hydrological conditions. I'd like to thank the multiple agencies and groups that have funded our work, including the BLM, U.S. Fish and Wildlife Service, National Park Service, and NSF. And lastly, the collaborators listed here who have been instrumental in helping with the work on this project. Thank you very much.